Hi, my name's Emily Cerda. I'm a microbiome specialist here with Tiny Health. Today I'm going to talk to you about digestion and absorption capacity metrics in the Tiny Health shotgun metagenomics gut test. So here on the main results page for the patient, I'm going to click into the big blue button that says See All Metrics. And then you're going to get everything here. A quick way to get to digestion and absorption is just to come to this left side menu and to check that box. So digestion and absorption markers here. You will notice fiber digestion capacity, complex sugar digestion capacity, protein breakdown capacity, which I'm going to do in a separate video. So go check that out in your practitioner resources modules. And then vitamin production capacity. So a common thread here is that we see the word capacity over and over again. And in Tiny Health shotgun testing, we're measuring DNA. So we're measuring who is in the microbiome, those are the microbes present, and we're also measuring what they're capable of. And those would be functions or proteins and enzymes that will code for certain gene functions of those microbes. So here, when you see the word capacity, we are looking at those functional metrics and we're looking at genes in those in the microbiome. So here in the digestion and absorption markers, the first section talks about fiber digestion capacity. So this individual's microbiome should have the capacity to help with all of these types of fiber digestion. If I click in to cellulose, for example, you'll notice that the green part or the optimal part of this metric is all towards the high end. So we want these genes to be present in the large intestine in that microbiome so that it's helping with the digestion of cellulose. If I back out here, whoops, I'm going to go right back to where we were. In the resistant starches. So we can see that this one is suboptimal. If I click in, now this is right on the edge, but the way I would describe this is to say that the microbes in the large intestine, they should carry the genes to help with resistant starch digestion. That microbial population has started to dwindle. That means the number of genes has also started to dwindle. And so the only way to kind of have more capacity or to grow this population is to feed it the food that it likes. So in essence, when you see something that comes up in the yellow or the red range, it means to eat more of this food type. Now, there's some caveats to that because as we lose these microbes and lose the genes associated with this digestive capacity, we also have reduced digestive capacity for this food type. So we can't go out tomorrow and eat all the resistant starches because the microbiome needs to build up that digestive capacity again. And if we did that, we may experience things like bloating and discomfort. So as you look at the action items associated to eat more resistant starch, you will notice some tips to eat this daily, but to ramp up slowly. So we're talking about small servings, and you can come in, you can look at the foods associated, you can bookmark if you wish. That we do have supplements as well, if that's easier for the patient. So this is about those microbes not being present that are carrying the genes to help with this function. Okay, back in to digestion and absorption. So these are the fiber types. Then underneath that, we have complex sugars. So these all look optimal for this patient. It is the same story as those fibers above. If we had something in the yellow, it means that we need to eat more of that food type. The isomalto oligosaccharides are something I like to point out because those are fermented foods. And so often, if somebody doesn't have those in the diet, or that not very many in the diet, but we have good kind of gene digestion capacity in the microbiome for this food type, I say let's add it and let's add it slowly so that we can really build diversity in the microbiome by getting some of that beneficial bacteria in there. And also, this function is already present, so let's go ahead and use it. Okay, I told you we were going to talk about protein in a separate video. And then down below, we have vitamin production capacity. So this one, these are actually postbiotics that are created by the short chain fatty acids. So I like to think of this as how well are your short chain fatty acids performing to create the nutrients that they should be in the microbiome. Here we see that the folate, the vitamin B9, is a little bit reduced. So the genes in the microbiome to create vitamin B9 are a little bit diminished. And in order to support that, we're looking at supporting short chain fatty acids. So first question for supporting those short chain fatty acids would be, are there beneficial bacteria? If we go back to this individual's beneficial bacteria levels, do they all look good, especially the Faecalibacterium that's really great at producing butyrate? And then if beneficial bacteria looks good, how is the patient's fiber intake? Because you really need to have both in order to promote short chain fatty acid production in the microbiome and therefore have them function well and produce these postbiotics. You will notice that the action items here say incorporate more vitamin B9 or more folate into your diet. It's really important to know that this is not nutrient status. Okay, this is a stool test. This is not a serum test. And by suggesting that you incorporate more B9 into your diet, it's potentially filling a gap where this postbiotic is not being made to the degree that it should be. It is not a hint that this person is a low folate status overall. So that's an important thing to recognize because a lot of times the patients will come in and they will see that that is out of range and they will assume that they are low nutrient status. So hopefully this has been helpful. Again, capacity is looking at the function of the microbiome. Is the microbiome able to perform these functions? And hold tight for that protein video. Go ahead and check it back out in your practitioner resources.